Hi, this is a short presentation describing the main findings from our recent study. The study is titled Peak Fat Oxidation is Positively Associated with Vastus Lateralis CD36 Content, Fed State Exercise Fat Oxidation, and Endurance Performance in Trained Males. This study has just been published in the European Journal of Applied Physiology. First, a brief bit of background. The peak fat oxidation rate, or PFO, is routinely assessed in non-invasive physiological profiling assessments undertaken by endurance athletes and is used as a marker of an individual's capacity for fat oxidation during exercise. Several high-quality studies have investigated the skeletal muscle determinants of PFO, with type 1 fiber percentage, mitochondrial protein content, and the activity or abundance of various proteins involved in fatty acid metabolism shown to be positively associated with PFO. Relationships between the fatty acid transport protein fatty acid translocase, cluster of differentiation 36, or CD36 for short, and fat oxidation during exercise have not been assessed. CD36 is an interesting protein to study in this context, as CD36 translocates to both the sarcolemmal and outer mitochondrial membranes to facilitate fatty acid import. Also, the utility of using PFO, typically assessed during fasted incremental exercise, and fuel utilization during training and competitive scenarios, as well as endurance performance, is not properly understood. Therefore, the aims of this study were to firstly assess relationships between PFO measured during a routine physiological profiling assessment, that is, a fasted incremental cycling test, and CD36 abundance, endurance performance, and fat oxidation during prolonged moderate intensity exercise in the fed state. The second aim was to determine if PFO explains any variation in endurance performance on top of that explained by traditional models of endurance. 17 endurance-trained male cyclists participated in this cross-sectional study. The participants performed an incremental exercise test in the fasted state, which started at 95 watts and increased by 35 watts every 3 minutes. Indirect calorimetry was used to calculate PFO. Approximately 48 hours later, a resting muscle sample was obtained using the microbiopsy technique. The abundance of CD36 in these samples was assessed using an ELISA and expressed relative to sample protein concentration. Whilst ELISA has disadvantages in complex tissue like muscle, it is fully quantitative, which was important in this work. Approximately 48 hours following the biopsy, participants reported to the lab again, having had a standardized carbohydrate-containing breakfast. They then cycled for 2 hours at 80% of the previously established first ventilatory threshold power output, therefore in the moderate intensity domain, during which time substrate oxidation was measured and glucose was consumed at 60 grams per hour. Following this 2-hour preload, participants performed a maximal effort 30-minute time trial with the instruction to complete as much work as possible. Participants were blinded to power output during the time trial. Now to our main results. As hypothesized, PFO was significantly, moderately, and positively associated with CD36 content. You can see in this case we used Spearman's rank as CD36 was non-normally distributed across the sample. However, fat oxidation during the prolonged moderate intensity fed state exercise prior to the time trial was not associated with CD36 abundance. As hypothesized, PFO was significantly associated with fed state fat oxidation. This relationship was strong with a Pearson's correlation of 0.83. There was also a strong positive association between performance in the 30-minute time trial that followed the 2-hour preload and PFO, with a Pearson's correlation of 0.76. To assess the relationship between PFO and performance that was independent of other classical markers of endurance performance, we constructed linear regression models. Our traditional model, which included VO2 peak in mils per kg per minute, power output of 4 millimole blood lactate concentration, and gross cycling efficiency, that produced an adjusted R squared of 0.877 and therefore explained approximately 87.7% of the variation in time trial performance. We added PFO in grams per minute to this model, and this increased variation explained to 90.3%. We also put these variables through a stepwise model selection process, which identifies the variables in the most predictive but concise model. This process removed gross efficiency and retained peak fat oxidation. Two further quick points on this slide. Firstly, I've not truncated the y-axis here to indicate that the contribution made by PFO to these models was indeed small, and also to marvel a little at how effective our long-standing physiological models of endurance are for explaining performance variation. Secondly, a criticism of our models may be the use of power at 4 millimole blood lactate concentration, and that maybe more robust measures of the physiological threshold may have been more appropriate. We considered this and ran our models with the more theoretically robust second ventilatory threshold instead. In this case, the same inferences were made. Inclusion of PFO explained a small amount of additional variation. However, the variance explained in these models was actually lower. So variation explained was in the mid 80% compared to up towards 90% with power at 4 millimolar. So in summary, one of the most interesting observations was that CD36 content was related to fasted PFO during incremental exercise, but not fat oxidation during prolonged exercise in the fed state. Whilst this work was only associational, it may be that CD36 is less determining of substrate oxidation in the fed state, where circulating non-esterified fatty acid concentrations and rate of appearance of those fatty acids is likely lower. PFO was strongly related to fat oxidation during exercise in the fed state. This means that we can expect athletes showing the highest PFO values during routine profiling assessments to oxidize the most fat during training and competition scenarios where carbohydrate feeding is more common. And lastly, PFO made a small contribution to variation explained in this model of endurance performance, i.e. a 30-minute time trial preceded by 2 hours of work, which could plausibly be due to athletes with greater capacities for fat oxidation being able to preserve glycogen availability during the preload for use during the time trial. Thank you.